So this mechanism is the glucose production was explained to the Melvin Kelvin along with the supporters. This Kelvin worked in green algae by using radioisotope carbon C14. C14. So here the Melvin Kelvin and along with supporters, co-workers explaining the whole pathway in how carbon dioxide, the CO2 converted to the glucose. So this Kelvin cycle, another name of the Kelvin cycle, it is the C3 cycle. Here there is a three steps. Here only the three steps is involved. The first one it is a carbon fixation, and the second one reduction, and the third one it is a regeneration of ribulose base phosphate. Here in this Kelvin cycle, one of the enzyme it's played a very important role, the enzyme we call Rubisco. So Rubisco, it is the enzyme. So in enzyme, the site is presented. The site it is a active site. So this site here I have highlighted in another color. The site we call active site. The carbon dioxide, the CO2 and oxygen. CO2 oxygen substrated in Rubisco enzyme, the CO2 and oxygen substrated have same active site. When CO2 concentration is high, it's binded to the Rubisco enzyme. The Kelvin cycle process is started. Finally, it give to the glucose. When the oxygen concentration is high, it binded to the when compared to carbon CO2, the oxygen concentration is high. It binded to the Rubisco enzyme. Here, the process is started to the photorespiration. So now we see one by one. So when oxygen concentration is high, it binded to the Rubisco enzyme. The Kelvin cycle, the C3 cycle process is started here. Here, this CO2 react with ribulose bisphosphate in the presence of Rubisco enzyme. It given to the 3-phosphoglycerate molecule. Again, this 3-phosphoglycerate molecule, what happened here? The ATP is involved. ATP is converted to the ADP. That means one phosphate molecule is released to form to the 1,3-base phosphoglycerate. So it means the process reduction is happened. So again, this one base phosphoglycerated, here NADP is involved. So the NADPH is converted to the NADP. Finally, the glyceraldehyde 3-phosphate molecule is formed. Again, this glyceraldehyde 3-phosphate molecule again regenerated to the ribulose 5-phosphate. So this whole Kelvin cycle it's run. Finally, it given to the 1 carbon. So normally glucose it is a 6-carbon molecule, but here in Kelvin cycle we got only 1 carbon. What about remaining 5 carbons? So in Kelvin cycle inside the plants it run in 6 times. So each and every time it is given to the 1 carbon. So after 6 times it run to the finally we got glucose molecule. So this is the Kelvin cycle or C3 cycle. What now we see the photorespiration.